don't know. What? Action.
Okay. Here we go. Mr. Mahoney here in our first YouTube video. I got my assistant here, Lily, and she's going to help in the Corn Inheritance Lab. So before we get started, here's what I'd like you guys to do. I have on my Chromebook, on my computer here, I have the MyCPS page pulled up for this week. So tomorrow will be April 6th. So make sure you guys click on the Corn Inheritance Lab and before we actually start this lab setup. And here we go. We're going to open up this lab. Corn Inheritance Lab. So just like in class, guys, I'm going to start by reading the background. What's so funny? Why are you laughing so much?
You're laughing.
Alright, we have the Corn Inheritance Lab. It says, you will plant corn seeds today. Why are you laughing so much? I don't know if I'll be able to do this with you. Alright, so you're going to plant corn seeds today that carry genes for producing plants that make chlorophyll. Now chlorophyll is that green pigment inside of the chloroplast that makes the plants green. As well, genes that lack the control of making chlorophyll. So you're going to have some plants that are going to be green, and you may have some plants that are albino. So you should have learned in your videos on mutations that albinism is it's the absence of pigment, and in this case the absence of pigment in plants is chlorophyll. So it says plants have the probability of carrying genes for making chlorophyll, and that's going to be our capital G. Will you hold that up for me? Okay. Plants have the probability of carrying the genes to make chlorophyll, capital G, or lacking this ability, and that's lowercase g. So in this case, the capital G is going to be dominant, and that's going to be making chlorophyll, and that's going to be a green plant. And then the recessive gene is going to be albinism, no chlorophyll. So remember, the rule for the recessive gene, the only way a plant will have albinism is if they have two recessive alleles. So they need two lowercase g's. Thank you.
Yeah, so.
Okay. The genotypes of the plant seeds could be homozygous dominant, so it's uppercase G, uppercase G. Am I taking too long?
No.
It could be heterozygous, it could be uppercase G, lowercase g, or it could be homozygous recessive, and that's two lowercase g's. After the seeds sprout, you will observe the phenotypes of the seedlings. So remember, the phenotype, pH, is the physical appearance of an organism. So in this case, the phenotype that you will physically see is the color. So a few things about these plants. So these are corn seeds. So why don't you hold on up, Lily? They look like just like a popcorn kernel,
Yeah.
but these are second generation corn seeds. And they've been genetically modified, and their parents, the parent plants of these seeds were both heterozygous. So, both parents were uppercase G, lowercase g. You can put it down. Thank you. So the materials for today, if we were doing this in class, you guys would each get your own cup, but here we are going to do our own little bucket, and we have rows. We have a row for period 1, period 3, period 4, and period 5. Okay, so if you guys look at your lab on the first page, you have independent, dependent, and controlled variable. Just a reminder, when you do controlled variables, you need 3 controlled variables. So please, please, please listen closely as we go through the setup. I'll be hinting towards these variables. So the independent variable, just remember this, it's the one thing that we are purposely making different. Okay? It's the one thing that we're changing. The dependent is kind of like the data we're collecting. And the controlled variables is we're going to try our best to keep everything the same in this experiment. So when you think about the controlled variables, think about things that plants need, because we're only going to make one thing different. Here we go. So I'm going to start planting period 1. So period 1, can I have 6 seeds, please? Yep. So what I'm going to do is I'm going to plant 6 seeds. And it says you want to plant them about 1 centimeter deep. 1, 2, 3, 4, 5, 6. There we go. So here you go. First period, you're going in. 1, 2, 3. Let's move this one a little bit.
You need an extra.
Yep. 4,
Need space.
5,
6.
and 6.
And
So,
then you do the next.
my assistant Lily will be planting period 3, 4, and 5 as I continue to go over the lab setup here. So, if you guys scroll down to page 2 of your lab, it's the predictions. And there you will see your Punnett square. So your Punnett square there shows the heterozygous parents. So they're both going to be uppercase G, lowercase g. So go ahead and do the cross. So if you forget how to do the cross, go back to last week and there's a few videos there on the Punnett square. Now, when you guys are asked about possible genotypes, now just know this, the genotype is going to be the genes or the alleles, so think of it as letters. So those are going to be the genotypes are the letters that are inside of each of the squares. When it asks for the phenotype, that's that physical appearance. So it's going to be the color of the plant. So let's talk about this setup here as she's planting. In there we have the same soil. They're each going to get 6 seeds. All of the corn are exactly the same except there's one thing different. One thing. And that one thing different is some of these plants are going to be green. And some of them are going to be white, but there's something that has to cause that change. So the one thing that's going to be different about this is the genotype. It's the genotype of the plants. Are you doing okay? Do you think you're going to be invited back to this next class? I told you, we've got one take. We're doing it. We're going to roll through with it. So the one thing that is different is the genotype. Everything else is going to be kept the same. We're going to keep the same soil. We're going to keep the same distance and the depth. Focus. We're going to keep the same depth. We're going to give the same amount of water. We're going to give the same amount of light. It's going to take a week or two for them to start to germinate, to start to sprout. So we're not going to know what color they're going to produce. Some of them are going to be green and some of them are going to be white. So that's all because of their genotypes. And you'll be able to figure out, thank you, you'll be able to figure out the percentage about what % will be green and about what % will be white. And then once we have them germinate in about a week or two, that's going to be our data. And what we're going to start to do for the data, the measuring, is we're going to count. We're going to count how many are green and how many are white. So make sure as you guys are going through, you do your variables. That's going to be a total of 5 points. 1 point for independent variable, 1 for dependent, and 3 for controlled, because you have 3 controlled variables. And then you're, thank you, and then you're going to have another 5 points for the predictions. So on page 3, there's your data table. So the number of seeds that we are planting for each class, I'm going to type this in right now, each class is getting 6 seeds. Now, multiple classes, I have 4 classes. Each one has 6 seeds. Lily, 6 times 4.
What?
What's 6 times 4?
24.
24. So there's going to be a total of 24 seeds for all classes. Now you'll notice in your data table, some things are yellow, and some things, I'll actually give you an example. If it's yellow, it says leave blank until the corn sprouts, because then we need to actually count. But there's one thing you can do. If I have 24 plants, or even for your class, if you have 6 seeds, you want to do a ratio. A ratio could be something like 2 to 2, 3 to 1. How many of them will be green to white, okay? And then that'll be it. You can actually, if you scroll down past the data table, you'll see, what do you see there, Lily?
Hand.
And
Stop.
stop sign. The stop sign means stop and then submit it, because we're going to grade these in stages. You will get graded for, where are you going? So you will get graded for the pre-lab, watching this video and filling this out, and then you'll submit it. And then once they sprout, we can then continue on the rest of the lab. So before we move on, I got a few things here. We will have our next Zoom. There's an email that went out that is explaining the different office hours that we can have. Now the office hours for us can either be a time where we'll have a virtual Zoom, or it's also a time where I'll be available for you guys to touch base with me and you'll get a quick response. So in this case, Wednesday, we're going to have a Zoom. So Wednesday, it is going to be the 8th, April 8th at 2. At 2 p.m. we are going to have a Zoom and we're going to review some of the pre-lab questions that you guys will hopefully fill out by the time that we meet at 2 on Wednesday. Now we have a lot of extra seeds, a lot.
And a lot of packages.
A lot of packages, because the original plan was you guys were going to work in groups and you were all going to plant. So I want to give you guys some corn seeds. So I will give corn seeds to the first 10 people, but you got to do something. The first 10 people that message me or email me with an answer to this question. Are you ready? Are you ready? Okay. The question is, why does corn always have an even number of rows on each ear? So next time you eat corn on the cob, count how many rows of kernels you see there, and there's always an even number. But there's a reason. So if you email me or message me on my CPS Classroom with an answer of why does corn always have an even number of rows on each ear, I will take some seeds and I will send them in the mail. Yeah, so that was our first video. I will say it's very different than teaching at school. I miss you guys. It is 6:47 p.m. on a Sunday that we're making this video. Okay? So guys, if you have any questions, you know how to reach me, email or my CPS Classroom Messenger. Pay attention to the YouTube videos and the messages that go out. So what do you, I never made a YouTube video. What do you say at the end? Bye?
Bye.
Alright, bye. Alright, let's go get it. And then we gotta hit the button. Okay, let's see. Hit that button right here.